In this talk, I'm going to be covering some of the work we performed in characterizing color peak springs and analog environments for the waters of late Noachian Mars. For the purpose of this talk, we are defining an analog environment as a site possessing a chemistry approximating that of a celestial body of interest. So in terms of an analog for the surface of modern-day Mars, you would expect an analog environment to have one of the following traits. It would be saline, oxidizing, potentially anaerobic or microaerophilic, and be exposed to high levels of ultraviolet radiation. Which of course highlights that an analog environment is an analog at a specific point in a celestial body's history and in a specific location. The geology of Mars is not homogeneous, so there is variation in chemistry between different sites. The surface of modern-day Mars is believed to be greatly different from the subsurface, and Mars 4 billion years ago is believed to be incredibly different and much more water-rich than its modern-day counterpart. Color Peak Springs is located on Axel Heiberg Island, which is in the Canadian High Arctic. It's characterized by polar desert conditions, meaning it's both very cold and very dry, and it lies within the region of continuous permafrost. The island is host to a series of sulfur-rich and saline cold springs, with some marked on the map shown here with either red dots or the gold star designating Color Peak. Color Peak is incredibly saline, like the other springs on the island, and sulfurous. But one delineating trait is it is also incredibly sulfidic, as evidenced from the black coloration visible in the sediment following from the spring source. This environment was worthy of further characterization from the microbiological perspective, because the microbes that survive and are active within this environment had previously only been partially characterized. And from the astrobiology perspective, this environment is argued to be an analog for Mars during the late Noachian. When we are considering analog environments, we first have to ask, is it chemically and physically relevant? No environment on Earth will be a perfect fit, but we want it to be as relevant as possible. In terms of the temperatures experienced by the Color Peak Springs, these fall within the range of colder temperatures that are modelled in predictions that estimate a cold and wet Noachian Mars, as opposed to a warm and wet Noachian Mars. We then analyse the chemistry of the Color Peak Springs using ICP-OES. Shown on the ternary plot, we have the three most abundant elements, calcium, sulfur and sodium, and in the bottom right corner we have here, in the red circles and the other circles directly on top, the different chemistries of the Color Peak samples, showing that sodium was the most abundant element. The other markings on this ternary plot designate other analog environments on Earth, such as the Deccan Traps in India. However, some also represent modelled Martian waters. For the purpose of this study, the program ChIM-XPT was used by Dr. Susanna Schwenzer. Using as input pure water and the chemistry of rock nest sand collected at Gale Crater by the Curiosity rover, ChIM-XPT estimates what would the chemistry of the solution be like if the minerals were to dissolve. And if we go back to this ternary plot and we look to the right of the red circles and to the leftmost side of the outer red circle, we see two diamond shapes, representing the thermochemically modelled waters based on these Gale Crater evaporites. So at least in terms of the most abundant elements, the chemistry of the waters and sediment of Color Peak are close to the modelled Martian water chemistries. The next question in our set of experiments was what microbes are present in Color Peak that are withstanding this chemical and physical regime. In order to answer this, we extracted the DNA from replicate sediment samples using a specially tailored DNA extraction technique to account for the high concentrations of salt within the sample. We then sequenced the 16S rRNA gene, the universal barcode gene that's found in all microbes, and it gave us the following pattern of diversity. The key takeaway from this bar graph is the dominance of purple and shades thereof across these three different bars from the replicate sediment samples. These represent sulfur oxidizing bacteria, and as is clear from these graphs, sulfur oxidizing bacteria dominate the color peak sediment. So who are the sulfur oxidizers? Sulfur oxidizing microbes are those that yield energy through the oxidation of reduced sulfur compounds, which we know to be abundant within the color peak and present on the surface of Mars and within Martian meteoritic materials. Sulfide oxidation can occur using oxygen as a terminal electron acceptor. It can also occur in the absence of oxygen using nitrate as an alternate terminal electron acceptor, which is also relevant in terms of Mars given the detection of nitrates within Martian mudstone samples as reported in the Stern et al. 2015 paper. Many of the sulfur oxidizing bacteria that we detected within the color peak sediment are also autotrophic, organisms that convert inorganic carbon, in this case carbon dioxide, to produce organic carbon, in the same way that plants do. They do not need, nor can they utilize, an external source of organic carbon, as we do as heterotrophic organisms. There is, however, a slight issue with identifying what microbes are present in color peak only by looking at DNA. And that is, in a cold and saline environment, DNA from long dead cells could hypothetically persist for millennia. In order to try and move past this issue, we also extracted RNA from the color peak sediment in order to try and look at what microbes were more genuinely present within this specific environment, as RNA has a much shorter half-life even under optimal conditions. Comparing a collapsed DNA profile to the RNA profile, we see that there is a significant reduction in the diversity present within the environment, and we see that there is this continued persistence both of the sulfur oxidizing bacteria, represented again in purple, and of some of the additional diversity within the sediment. But the active microbes are still mostly sulfur oxidizing bacteria. The question is then, independent of the microbiology point of view, why is this an interesting result? Why is it interesting that microbes with this specific metabolism are living and thriving in this cold pool in the Arctic? And moreover, why do we perform any kind of characterization of analog environments? And the answer is, it enables us to generate hypotheses. We know that a lot of the diversity detected within this sediment of this sulfur oxidizing bacteria are capable of autotrophy, meaning that they can fix carbon dioxide as their sole carbon source. Combined with how carbon poor the sediment is, they're most likely surviving within this environment through the fixation of carbon dioxide. However, as these cells are growing, some will be leaking, either intentionally or unintentionally, and as some are growing, they will be dying, releasing a pool of organic carbon that they themselves cannot utilize. And we know that there is this additional package of diversity within the color peak sediment. It is therefore possible that the other bacteria within the color peak sediment are surviving by growing on the carbon produced by the autotrophic sulfur oxidizers, and this is something shown to occur in other environments such as hydrothermal vents and also in cultivation-based studies. Now, in terms of this metabolic process, we know the concentrations of the individual elements that are key for sulfur oxidation. And the same is also true for Martian conditions. We know the concentrations of nitrates detected within the mudstone samples, and we know the thermochemical composition of the waters that were derived from the chemistry of the Gale crater evaporates. Using Gibbs energy equations, it's possible to deduce whether, based on the abundance of the elements that are required for specific metabolic processes, whether there would be sufficient energy yielded to make this a thermodynamically favorable metabolism. And doing these Gibbs energy calculations, these calculations identified that not only was aerobic and anaerobic sulfur oxidation feasible under these proposed Martian conditions, they were two of some of the few metabolisms that were indeed capable in this specific chemical environment. So how can we take this one step further? How can we test if the analogue community is viable in genuine Martian chemistries, moving one step beyond the characterization of the proxy environments and the modeling of Martian chemistries? And we can do this through performing simulation experiments. If we take one step back to the CHIM modeling of the rock nest sand chemistry and the pure water, we can use this output to produce a liquid medium in which to challenge cells. We can then combine this with two different chemically accurate Martian regolith simulants, one based on the chemistry of rock nest and one based on the more iron 3 enriched chemistry of hematite slope, establishing two distinct simulated Martian chemistries, performing this under anaerobic conditions in combination with the color peak sediment to more accurately simulate Martian conditions. The question is then, is the analogue community that we're arguing is viable under a proxy Martian condition viable under simulated conditions? And do we see the persistence of these sulfur oxidizing bacteria? If we look at the general microbial community again and compare this to under rock nest conditions, we see that the answer is indeed yes. The purple persists, indicating that sulfur oxidizers can grow under these chemical conditions. However, we do not observe this under the iron 3 enriched conditions of hematite slope, where the sulfur oxidizers are in fact eliminated and substituted by these sulfate reducing bacteria, with the reasons why requiring further experimentation. In conclusion, color peak is an appropriate analogue for Mars in the late Noachian, and it's dominated by sulfur oxidizing bacteria, a metabolic process that is feasible under proposed Martian conditions and viable under simulation conditions, but only some.